So good morning, and to our friends joining us abroad, good evening and welcome to Merage Talks. Today we invite you, our talk is Cell Culture Cards and NASA's first GENESAT space mission with President and CEO from ALine, Dr. Leanna Levine. Thank you so much for taking the time to join us this morning.
All right, thank you. So I appreciate the opportunity. I'm pleased Ari invited me to give this talk. I'm going to talk about work that kind of goes back to the beginning of the business back in 2003 and talk a little bit not only about this particular project with the GENESAT, but some other work we've done with NASA, as well as a little bit about how I've grown the business and the ways that we've been able to network and grow our engineering services business. So a little bit about me. I'm a PhD bioanalytical chemist. I started ALine back in 2003. This was after having been in both large companies, small and startup companies. Always wanted to start my own business. I had the idea that people needed a way of getting high quality microfluidic prototypes, which I'll talk a little bit about microfluidics in a moment. And so the premise, the mission for the business was to offer a rapid prototyping technology, a platform technology we developed in-house, and then to offer that to customers so that they could focus on developing the biology and the test within the system while we provided the high quality prototypes that would enable them to do that. And that really is what I'm going to talk about with this set of case studies around the NASA GENESAT mission. So on the next slide, what we do is we really focus on, I guess, what you would call the translational science and engineering. Many of our customers have some really cool technology that they developed in the university, and they're looking for a way of commercializing it. And the thought processes that go into what you would do in a research lab versus what you want to do to develop a product are quite different. So we spend a lot of time up front helping our customers to think about the right ways to mitigate risk and to embark on a development program that's going to get them to proof of concept and support their ability to get further investment to push towards commercialization. So the 4 areas that we work with customers with our microfluidic platform is in the development of single-use diagnostic cartridges, typically for point of care and rapid testing. I will say we have many projects focused on COVID right now because there's been a lot of investment by the government as well as a push to deliver these kinds of tools to businesses, individuals, so that we can help to control this pandemic. Secondly, we work with companies that are developing more sophisticated cell culture systems. They need multiplex fluid handling, which is something that microfluidics is very good at supporting. And then we also work in developing some approaches to cell culture and a new area called organ-on-chip development that is changing the way that pharmaceutical discovery is happening. And then finally, we have had some long-term customers that are involved in developing wearables, sweat patches for athletes. Originally, the work was supported by the Air Force Research Lab. It was really to monitor soldiers so that they would get an indication of when they ought to be drinking water or replenishing their electrolytes before they collapse on the field. So these have all been involved a lot of different problems to solve, ways of thinking about solving them. And of course, any business needs to have a good team of people that help us make those things happen. So in addition to myself, I have a director of engineering, Stefano Bagolo, who's been with me for over 5 years now. He came out of Caltech. He did his postdoc there. He got his PhD at the Marie Curie Institute in Paris. And then we've recently hired another PhD out of Caltech, Eric Ju, who is also in the same group that Stefano is in. So we're able to do what we do because we've hired a really high-powered team of engineer scientists that can help our customers deliver on their goals. So first I wanted to talk a little bit about, well, what is microfluidics? And if you look at the images on the left, you'll see someone using pipetters and test tubes and microfuge tubes for manually handling small volumes of fluid in order to do a biological assay, whether it's something like PCR or an immunoassay. And then what you see also are systems that rely on the use of tubing sets in order to move fluids from one place to another. And so these are some of the more traditional ways of doing science that have been done for the last 50 years. But with microfluidics, it's driving more towards taking all of this functionality and putting it in a small plastic cartridge that allows the operation, all the operations that would have been done manually to be done on a small fluidic card and operated by an instrument. So, for example, all of the tubing that you see here and fluid delivery can actually be controlled by this fluid delivery manifold that is part of a commercial product that we worked with BioLab Technologies to develop. So microfluidics is, I think, what I often tell people, it's tiny plumbing in plastic. And just as you think about plumbing networks, they're three-dimensional and they move from one level to the next. So we're taking and stacking fluidics in these fluid cards in order to get a lot of sophistication in a very small area. So why do we want to do microfluidics? And I think I've alluded to that as you can take manual operations and automate them. But if you think about the traditional ways, for example, of doing cell culture, none of these technologies would really be amenable to doing things in space. They need to be self-contained. They need to be managed in an automated fashion without too much power and intervention from people. So microfluidics enables that. It miniaturizes. Because of the dimensions are so small, you get novel performance because of the small surface area, the large surface area volume. You can do things in a massively parallel manner. So you can collect lots of data in the same period of time, which really supports now our ability to outwork big data. And then that, of course, drives low cost data points. So the amount of information that you can get from biological systems and from biological tests has just exploded with these, with microfluidic capabilities. So let me dig in to the GENESAT program itself. This was a program that was started back in 2003, which was the same time that I started ALine. The reason I was introduced to this opportunity was, of course, I had a pretty large network of colleagues that I had worked with when I was in industry. And one of them, Tony Rico, had moved to NASA Ames in 2003. And he was hired to work with this gentleman, John Hines, who was the director of the small payload program and was responsible for a program that was aimed at being able to do biological experiments autonomously in near Earth orbit. So this would eliminate the need and the time and expense that it takes to do a biology experiment on the International Space Station. So to do something on the International Space Station, it takes about 18 months of planning. It costs at least $250,000. And it's difficult to get too many things done because it requires human intervention in order to make it happen. With these small autonomous satellite experiments, they're able to do sophisticated biology experiments without any human intervention. But the key here is, how do you take all that sophistication and package it into a small satellite that's about the size of a loaf of bread and have it perform and record the data that you need. And so primarily, the initial experiments were focused on understanding what are the effects of weightlessness and radiation on living systems. So these are two of the things that we know. I'm sure all of us have followed different aspects of the NASA space program. When astronauts are on the space station, they have to exercise regularly because their bodies are not experiencing gravity and their biology changes. They lose bone mass. That's one of the large things that happens. And so how could we study these effects in small systems on autonomous satellites? And what we would learn there could help us to figure out how to do further space exploration with living systems, but also inform how we can improve the therapeutics that we use on Earth, say for patients who have osteoarthritis and benefit from changing the biology of the system in order to eliminate that disease. So these satellite-based biology experiments have opened up a whole new field of science. It allows an experiment now to be done on a satellite package that costs about $40,000 versus $250,000. So this opens it up to university groups. And over the years, we've had interest from different universities to help them design their own fluidic cell culture card to do their own experiments in space. So what are some of the challenges? And I alluded a little bit to that. On the satellite package, these loaf of bread sized satellites need to be low mass. They have to have low power consumption. They need to be ruggedized so that when they get shot in a rocket, they don't break. And at the same time, once they're launched from the rocket, once they're in low Earth orbit, they need to open up, come to temperature, and then start the biology experiment. So that's a lot of sophistication to package into a small system that has the instrumentation, the optics, all the components that are needed to operate. And so here on the left is a picture of Chris Beasley. He was one of the scientists that we work with at NASA Ames to develop this package. And here on the right, you can see how the circuit board here that contains the cell culture card is sitting on top of this unit with the optics, and then this all sandwich all comes together and then gets inserted into this tube. So to talk a little bit about what were the fluidics challenges for this system. And I have, once again, there's kind of a schematic picture of the package that gets inserted into that cylinder. It contains, you can see along here is the optical rail that's used for interrogating each one of the wells in the cell culture card. All of the heating and the circuits to drive the system. And then over here on the right, you see a container here, and inside of it is it's an IV bag that contains the growth media, and from that are just two tubes that lead to the cell culture card to provide the growth media and then to remove the stasis media into a waste containment area inside of the system. So how this experiment works in the cross section for this is the cells are loaded into each of these chambers here. So there's 10 of them. Yes, 10. And these cells are non-adherent E. coli. They've been engineered to produce green fluorescent protein once they start growing. And so in order to make this work properly, the stasis media has to be displaced through this channel, through a porous membrane, fill and replace all of the media in this chamber, and then exit through an upper channel through another membrane so that all of these non-adherent cells stay located in this area. And then the stasis media gets moved from here into a waste region contained over here. So the cell culture card needed to enable gas exchange in the surrounding area inside of that cylinder, would be pressurized and have the proper CO2 and oxygen in order to support growth. And on the opposite side, there needed to be an optically clear acrylic material to allow optimal interrogation of the system. So another key aspect to this, with a single inlet and a single outlet for the delivery of the growth media, was to start the experiment at the same time across all 10 of these wells. And in order to do that, these membranes really have a dual function in this system. And this was the innovation that we did at ALine, was to put a fixed area membrane with a known porosity that would act as precision resistors in parallel, so that when you bring fluid in here, it fills across all 10 of these at the very same time. So the experiment starts in all 10 chambers at the same time, so that they can do comparison experiments directly. So you fill through these channels, it goes across, this replaces the stasis media, and then exits through here into the waste area. So we were able to provide this kind of a sophisticated cell culture card because we had a platform technology that enabled us to place these membranes and not have them leak, and to have the area of the membranes inside of this fluidic card mapped well enough that you could get the desired filling across all 10 chambers at the same time. So this was enabled by our, like I mentioned, our platform laminate prototyping capability, which over the years we've evolved, and it's now not just prototyping, but we can scale it for manufacture and also have design rules that enable us to do roll-to-roll manufacturing for low-cost, high-volume point-of-care products. But the original technology was a rapid prototyping based on laser cutting and lamination. So it's a true prototyping technology, in that there's no tooling required. All of the designs are done in CAD system. You think about what the functional requirements of the microfluidic need to be, what the volume requirements are, and then you split the three-dimensional design into layers, and then machine cut each of the different layers to be able to get a final stacked fluidic card that you see in kind of in cross-section here that enables the functional performance that I described in the previous slide. So this was really the foundation for the business and for the technology platform. We've added a lot of sophistication over the years. Not only can we be able to place membranes with good quality into these systems, but we've also developed onboard pneumatically controlled valves and pumps and lots of other functional requirements that are needed for a lot of fluidic systems, particularly for doing sophisticated workflows for biology. So this platform technology and being able to do it with a level of quality and repeatability that was needed for the NASA program is what allowed us to successfully deliver for this program, and then for a follow-on program that we did a few years later. So the next program that we were involved with was what was called the Power Cell, and it was part of a larger European mission called Eucropis. Eucropis was interested in growing crops in space, understanding what weightlessness would do to growing food. So the thinking now has advanced to not just being able to understand what happens to living systems in space, but now how can we actually move living systems into space and onto different worlds. I'm sure you've all seen the movies, Martian, and even 2001 Space Odyssey. One of the things that this particular power cell system had to do was to simulate the gravity on Mars and the moon, and of course Earth. And the way they did that was exactly the way they did it in 2001 Space Odyssey, which was to take the system and rotate it and use the centrifugal force to create gravity. So science fiction movies and science fiction in general really does their research when they come up with these ideas on how to do these things. It just takes the real world some time to catch up. So this shows the team at NASA on the left. This is Carolyn Ranzano, who we worked with, and some of the other team members. They're demonstrating how they worked to get the cell culture card that we developed with these guys. Now you can see that the next generation of this device, instead of having just 10 chambers, now has 48. So the level of sophistication has gone up. And on the right, there's a picture of Daniel, one of our engineers who was responsible for this program. He really worked very hard to deliver over 200 different cell culture cards for this program. And once it launched in 2017, I believe it was, NASA had a whole bunch of these cards left over, and I got a picture of these cell culture cards hanging on a Christmas tree as ornaments. So that's what happens when after a successful program. So here I have a really cool video that they shared with me that describes this power cell project. And this, by golly, this thing better work, because it just worked before. There we go. Okay, that's a very energizing video. It's a tough thing to follow, but I think some of the things you'll notice that they featured the SpaceX rocket. And I'm sure you're all aware that the investment in private rocket launches also enables more exploration of space. The first GENESAT actually went on a Minotaur rocket from the Air Force when they were launching satellites for military purposes. But now more of this is moving towards being accessible for commercial applications. So it should continue to open up the field to more and more types of experiments like this. So another program that I wanted to just briefly touch on in another area that NASA is exploring, and another place where microfluidics makes a difference is in trying to learn and understand their signatures of life in our solar system. And one of the programs that we worked on at JPL with Peter Willis is an analytical system that would go on the Mars rover or on another rover that would eventually make its way to Europa, which is one of the moons of Jupiter, which would actually go probe the surface, below the surface, to collect a soil sample and then process it using a microfluidic system that would allow them to chemically alter it so that they could determine, are there amino acids, which are some of the building blocks of proteins, and if there are amino acids, maybe many of you know there's two stereoisomers. Life on Earth uses the enantiomer of the amino acids to build proteins, and that's driven by enzymatic reactions that come from living systems. So if you probe other worlds and you find that you do not have an equal amount of L and D amino acids from a soil sample, then you know that that would be a signature of life. So part of that, what we did working with Peter Willis's group was to develop this chemical processor. And what this allows you to do is to take inputs from any of the number of reservoirs and move it around in this series of onboard magnetic valves and deliver it to any output. And what I'm demonstrating here in this video is we're able to use the design for a valve system that would enable an actuation protocol. And as you can see, there's leaking that occurs between this valve and these adjacent valves. And this is an undesired performance characteristic. So we had to think about how we were going to solve this problem. And solving this problem wasn't going to happen by changing the design of the microfluidic or the design of the valves. We were kind of stuck with what we had there. So what we recognized was that we had to change the way we did the actuation of the valves and the way we control the pressure in the system in order to prevent leakage to the adjacent channels and valves. So I like to use this as a demonstration that it's not just the design of the microfluidic that's important for the performance, but it's also how you control it with the instrumentation. So we've spent a lot of time in the last 17 years really understanding not just how to do rapid prototyping of microfluidics, but to build a toolbox of functionality that allows us to do really sophisticated things that can operate in interesting applications, all the way from space to point of care and drug discovery applications. So I also wanted to highlight that because of this push and the ability to do commercial spaceflight, there are companies, and this is, I think, one of the only companies that I'm aware of, and they were one of our customers, was Space Pharma, which is actually an Israeli company that is, their mission is to have miniaturized microgravity lifts that are available for people to use to do the same kinds of experiments that I've just finished describing that have been done by NASA. Is the sound still bad?
From time to time, yes. Sometimes it gets better.
Maybe I don't have enough bandwidth.
It might be because now it's much better. Maybe.
Okay, so no videos, so that's probably helpful.
Oh, okay. But it was understandable. I mean, it was. I was able to listen. I hope others.
Okay. All right. So I've only got a couple more slides here. I'm sorry about that. It's hard to know how this will actually work when you've got videos. So I guess, in summary, the kinds of work that we do as contract developers is to develop miniaturized analytical systems that enable the evaluation of living systems in exotic environments, to develop distributed and localized testing with different types of point of care products. And then, of course, you can think about ways of linking those together to get centralized data collection and analysis. Some of these types of sensors for biological tests can be linked to other Internet of Things applications. And I think one of the things that I really like is the fact that these point of use or point of care products can enable the citizen scientist and allow people to collect and share information that supports them educating themselves and working as a community. So it democratizes access to science and information. And of course, kind of the dark side of this is that it, of course, enables surveillance capitalism, and because there's this data stream that's available for parsing across the population. So I think the way that we would think of how we support our customers is we help them to conquer the complexity of developing these systems because of our experience and our ability to focus on what is important for developing a product. So all of the things that help our customers be successful is our ability to deliver on a tight timeline, to create the structure and to identify the key technical risks and address those early, and to offer a toolbox of functions that enable that capability. And of course, being able to execute on a rapid design, build, test cycle so that the program moves forward quickly. So, and of course, with that comes being able to design for manufacture and to do pilot production. And as a company in this sort of a rather niche market, it's good to be well networked with industry experts and suppliers so that we can help our customers get access to the volume manufacturing and the supply chain that they need. So I would invite you to visit our website. We have a number of case studies that talks about the different kinds of programs that we've been involved in. And it's, for me, it's been a passion because not only do I get to stay at the cutting edge of science, but we also get to solve really interesting problems and different problems. So it's never boring, and we always learn new things, which means we have new things that we can then pass on to new customers because of what we've solved in the past. So just as a closing out, I invite you to contact us if you have any applications that you're interested in that need microfluidics or some sort of fluid handling capability. And I appreciate your attention for this talk. And I'm sorry about the sound. I could not have foreseen that one.
No.
So are there any questions?
I have some.
Yes.
Um, how many iterations usually are going in in inventing a new thing, a new, I don't know what to call those plastic things, or the whole process? I mean, looks to me like the focal point is innovation in each and every project. So just kind of maybe you can, I mean, we all struggle and maybe challenged by thinking out of the box sometimes and facing a problem and saying, okay, how do I tackle it? And I was wondering if you have some pointers there for the, for the group.
Well, so especially for medical device development, and a lot of this is driven by FDA. The thing you need to do is kind of work backwards. So you have a vision for the product, and you know how a person should work with it, and then you think about you develop a design control table, and then you think about all the ways you could screw it up. And then you think about all the technical aspects that you haven't quite solved from the bench to being able to do it as a product. And so the number of iterations, and this is where working with someone who is, you have a platform technology and you've exercised it on a lot of different programs, it's not difficult to get to a functional fluidic system, but it's really then the optimization and transitioning very quickly to a system engineering. So you have to optimize not just the microfluidic, but the biological assay that's being performed, and then how it interfaces with the instrument that controls it. So it's kind of a round robin in design, build, test. So you have to modify the assay, modify the fluidics, optimize the instrument interface and actuation. And that process typically takes 9 to 18 months, which is a broad range, but that depends on the complexity of the system and the sophistication of the workflow. And once you achieve that proof of concept, then the next step is, how do you then think about turning that into a commercial product? So you've gotten everything to work, and now you really need to focus on how you're going to move towards the commercialization. And so that brings in a whole other round of development that's needed to get it ready for transfer to volume adoption. So it's and that transfer process can take another year to 18 months. And then commercialization can take another few years. So, and I think this came from an earlier Merage talk that I was participating in. This whole process is about 8 years and, $30 to $50 million, if not more. That was a long-winded answer.
No, that's very good. Thank you. Thank you.
Leanna, I have a question, if I may. This is Eli Tionkin. What do you use for storage and compute in your bread size shape model?
What do we use for
storage and compute? So to run the experiment, I imagine you have some type of program, execution, application, container that takes the data from the storage or from the sensors that arrive to your system. So my question goes, how is the system handling the storage and compute in those LEO small satellites?
All of that is contained in the microprocessor that's telemetering the data back to Earth. So all of the data gets transmitted back to Earth.
Okay, I understand. So you don't do it locally in the small satellite, but you transmit it and you do those computation on Earth, in the ground station. Okay, thank you.
Are there any new research like that with NASA that is going to send some other biology to space to explore the agriculture?
I have not followed up since the power cell and the Eucropus. Unfortunately, the Eucropus, the crop part of the mission didn't work. There was a software update that caused the Earth-based computer systems to not be able to communicate with the satellite. Not the power cell worked, but the German the crop system didn't, all because of a mismatch in the updated software, which is
Wow.
shocking. Yeah.
Unbelievable.
Yeah.
So just one, I do have one more question. I don't want to dominate. Anyone else has a question, that's fine. But you know those, you say that you do a lot of on CAD, right? You design those channels. Does that give you ability to simulate as well? Or is it just for the design, or do you actually simulate the flow on those systems first before you build?
Yeah, so there are software packages that allow you to simulate the fluid flow, and we don't do that in-house. There's lots of folks that do that sort of computational modeling, but we have used it on a few programs, but in the end, we find that you have to just get the results empirically. So, and sometimes it's actually the design changes you make based on your experience get you closer to the desired functional performance faster.
Sure.
People are
Thank you.
still useful in the process.
That's good to know.
I don't know how long that'll last, but
Well, do we have any other questions for today before we close up? Well, Leanna, thank you so much for taking the time today. That was excellent. And for anyone who missed any portion of today's Merage Talk, you can find it on our site, meragetalks.merageinstitute.org. Please feel free to share with your friends, family, and colleagues as well. Leanna, thank you again. I appreciate it.